So today our speaker is Dr. Zhugen Yang, a principal scientist from UK CEH, and he's leading on the UK RI-sponsored National Wastewater-Based Epidemiology Monitoring Program to give us part of the first call from the UKI to support the information for government. So the methodology is try to detect the RNA sequence from the wastewater in order to provide the useful data for the intervention. So now over to you, Andrew. Looking forward to your talk.
Yeah, thanks very much, Zhugen, and thanks for inviting me. And I see there's a lot of familiar people in the audience, and half wondering if I'm going to tell you something you don't already know, because actually some of you have generated this data. But nevertheless, you're here, hopefully eating your lunch and maybe catching up on your social media. And in the meantime, I'll try to tell you something that you don't already know. For those of you who don't know anything about what I'm going to talk about, great. Thanks for coming. So I'm going to give you a bit of a perspective about the kinds of work that's going on on the national level in the UK, and then some project level work, and then dipping into the kinds of things that probably relate much more directly to a bioset or a sensor focused group at the end. I should say that I'm not a sensor kind of lab, where I don't run one. I just very much admire the people who create them. So for those coming to this very new, I'll start at the very beginning. So during a pandemic, one of the most important things to know is who's infected and where are they. That's a really costly question to answer using all the techniques that we've had available to us society, for tens to hundreds of years, which is surveillance and testing, and where there are, molecular tests, then it gets costly, and someone's got to take the test, and they've got to run it over to a lab, and logistically, it's a nightmare. So what you would ideally like to have, at least to complement that, is a method to know, what's the level of infection in any one location. And you would like that to be unbiased, because when you ask people, can I take a sample from you, they have the right to say no. So your ability to know and what is the spread of an infection in any one area can be quite biased using the current methods. So we're looking for an unbiased and ideally a low cost method. There would be no great benefit if it was not low cost, because there are trade-offs to having an unbiased sample of the nature that I'm going to talk about. And then you obviously need it to be easy to implement, so it can be obviously more complicated and more logistically challenging. So to that end, wastewater-based epidemiology is born. To be fair, it wasn't born during the pandemic. It existed for many decades, to be honest. But it was probably most effectively used for polio surveillance, which it still is. And in fact, someone on this call has done it, and it does do it. And what it, in principle, allows you to do is it allows you to understand all the people who are infected, who are shedding the virus. Then that material goes through the sewer system and to your wastewater treatment plant. At the point of collection, you then have the capability of knowing those who are symptomatic, those who are asymptomatic, and those who are pre-symptomatic. So everyone who is infected is shedding the virus, just to different degrees, and that's where some of the many challenges lies in trying to make sense of this kind of data. There's no shortage of challenge. Okay, many people on this call are actually part of what is called N-WESP, but the long version is National COVID-19 Wastewater-Based Epidemiology Surveillance Program. It was funded by NERC right towards the start of the pandemic, and it has evolved a fair bit over the months, actually years, and what it started off being able to do or proposing to do was to develop field and lab methods to collect, transport, store, extract, analyze, and interpret this data from wastewater. At the time we proposed this, there wasn't a national program, but there were some of the genesis of each of the nation's programs basically starting at that same time. And so they all needed the best evidence for how to do this. And this, the team, which are in black dots across the map, were the core teams that were feeding into that evidence base. Second thing that it was aiming to do was to look at the hazards posed by infectious virus. And at the beginning of the pandemic, again, there was a lot of unknowns, and there was no evidence as to whether infectious virus would be present in wastewater. And I'll get into that a bit later. Third one is to develop models to estimate the cases. So as I say, the amount of data that you collect it could be massive, but actually translating it into how many people are infected is not a trivial task. And so there were lots of efforts to try to do that. And the last one is the fate of the virus in wastewater was an unknown, and that was one of the tasks of N-WESP, is to get to terms with if when you release the virus, how much of it should reach a sampling point, maybe at a sewage plant. Is it 90%, or is it 0.9%, or is it 0.09%? So at the start, these were the contributors and the partners, and that team has grown hugely over time because of, well, a pandemic does attract a lot of attention, but these were the core members, and there's about 60 or so members who, to varying degrees, attend the weekly meetings that we have and share best practice and any innovations that we've had over the span of time. So I'll jump right into some of the evidence. I'm not going to, well, I suppose the evidence is what I'll be telling you. So one of the really important things that was greatly in question at the time of the start of the pandemic was whether this SARS virus remained intact after it being excreted, and then whether it remained intact as it transported itself through sewage, either into a sewage treatment works or into what we call CSOs. These are the sort of bypass valves of sewage works or sewer systems, and raw sewage would then go into the environment if those were triggered. And so it was of great interest to know whether those who work at a sewage treatment plant are at risk, or those surrounding it, or those who interact with CSO impacted rivers. So at the time, we still had a bit of a few months before the summer where people do use the rivers, and we wanted to know if that was a risk factor. The short version of this is that despite or because we've had a lot of people try to find the infectious virus and we haven't found it, we have quite a lot of confidence that there is a very low likelihood of having an infection from exposure to wastewater that had SARS in it. It doesn't mean it's impossible. It just means that you had a very unusual scenario that allowed it to happen. So that was actually really good news. And I'll move on to what is happening on a national level. So as I said, N-WESP fed into actually all of these programs, and the English program started off at a pilot of 4 locations, and it's over 84 sites now. There's about 500 plus locations also being where surveillance had occurred or is occurring. The aim was to have enough surveillance in the England to have approaching 80% of the population being under a degree of surveillance for COVID at the sewage works. Scotland had a similar aim, so they have somewhere approaching, well, I think they were aspiring for 100 sites. I think they're probably close to that. Wales, I think they're roughly at 20 sites. It may not still be going on, but there are some people on this call who actually carried this out. And Northern Ireland got off to a delayed start compared to the top three, but they have a similar aspiration for roughly 70 to 80% of the population being under surveillance. So that is what the national programs roughly look like. I have to say that it is a dynamic thing, so if it really is different from that, that's the last I know. Scotland's data you can access. It's on the web link below on the bottom right, and so you can see each of the locations that are under surveillance on the top as columns, and then the dates from which they started surveillance and then continued are reflected in the color of the slash or the bar. And so the more green you have, the less COVID, or in fact no COVID, and any other color than green is indicative of SARS was found in that sample to some degree, and if it's purple, then it was very much found. And well, it speaks for itself. There's a heck of a lot of purple in Scotland, and this is up until mid-September, this data. So there is no online report for Wales or reporting system for Wales, nor England, but there is an online report for Wales, and it was I think that it was August data that was most recently available, and you could see that there was a creeping up of cases in Wales at that time. This is on the left-hand side, it's April. In the middle is June, July, and on the right-hand side is September. So it's quite a significant increase, and those are the locations that were under surveillance with the web links on that page. So the surveillance was not just at wastewater treatment plants, and although the wastewater treatment plants are logistically a lot easier because a lot of them are manned by the water industry themselves, they have a lot of space that you can put a sampling equipment there. You could plug it into a power source, and it's secure, so there's no risk of it being tampered with. But it's not the only place that there is an interest in knowing the cases, the infection rate. So there are some really big cities, obviously, in the UK, and London being one of them. And so there is a desire, and there was a desire during the pandemic, to break up cities to smaller constituent parts. There was a sort of a pragmatic decision to sort of do it based on higher risk areas, so where some of the more traditional sampling was indicating there may be hotspots, then they would go in and find a manhole cover that was accessible and take a sample from there multiple times a week. And that data would feed into the national understanding as to what's going on, in this case in London, and then this happened across the UK in several cities. And so that's in a location that is not a wastewater treatment plant, which means it's constrained by all the attributes of a location, such as the manhole cover can often be in the middle of a road, which either means you need to stop traffic or you need to pick a different manhole. And so you can see that there's just that would constrain a lot of what you might consider on paper to be an ideal sampling location, because the branching points of a sewer system would allow you to capture some people in one location better from a particular node in the network. So there's a lot of sort of doing your best in these kinds of situations. Second big thing is that they were all samples were only taken by grab samples. So they basically just stick a bucket down, grab a sample, and then take it away and analyze it. So it's not what we call a composite sample of sewage over time. So it really is whoever flushed the toilet within the past 10 minutes is probably within the bucket that you just collected. So it's indicative, but it's not what you would like to rely on if you had your choice. We didn't have a choice because we sort of this whole system was invented on the fly. And so this is kind of what it would look like in a reporting sort of way. So you would have different parts of a city that are separated by a single node in the sewer system, and then you would basically just color each part of the city based on the amount of virus that you would find there. And then you would compare that trend over time. So now we're getting down to a sort of a project level. There's wastewater treatment plant surveillance, there's in the system, sewer system surveillance, and then there's near source sampling or tracking, where you're sampling outside of a building or sometimes inside of a building. And this was first demonstrated to be a useful approach at the University of Arizona, where they painlessly stuck a bucket down a manhole cover outside of a dormitory and kept taking samples and then analyzed them and found that there were infected people who had not been identified previously as infected. They went in, found those people, and reportedly stopped an outbreak. It happens all the time now, but this was the first reported example of using wastewater to identify people who were asymptomatic. So that was a really useful demonstration of what became something that everyone across the world was then going to do, which was surveillance of really important locations. And so the TERM project, so we have some people on the call who are from there, and the TERM project is originally set up to look for schools, and really it was a proof of concept, proof of principle project, because it hadn't been achieved here prior to this project. And it was just to demonstrate, can you get useful data out of schools? And originally it was set up to look at London, Bristol, and then at the time we set it up, which was almost this time last year, was the north was really quite looking bad as far as outbreaks. So we went to one city or small village, I guess, in the North Pendle. I'll basically have to say that the project went until Christmas, at which point there was a lockdown, and schools then had a varying amount of attendance. And so some of the extra capacity that we had, due to the fact that some of the schools weren't worth doing surveillance at, some of that effort was used to go to prisons. And prisons were a really interesting case because they are almost, in some ways, the opposite of a school, because everyone stays in a prison. You know exactly who's there all the time. Whereas schools, there was no obligation for the school to tell us who was there at any time. But we could tell them, in theory, who might be infected. We had a pilot project at a care home, and that was another proof of concept study. So I'll walk you through some of that data. So the first bit of data is on the run up to Christmas. And so as schools got onboarded, then more data started to show up. So the schools are in the rows and the columns are the days, and the frequency of sampling obviously gets better as the project ramped up into full steam. But it also coincided with the outbreak that everyone knows was the second wave during Christmas. And so by the time Christmas came around, roughly 80% of the samples that were being acquired were testing positive. for SARS. So it was clear that pretty much every school child was being exposed to SARS, and no one was being sent home because of symptoms. So they were presumably asymptomatic at that point. So it means that the asymptomatic carriage in that population was clearly very high. So this has changed. In fact, my daughter just got sent home this week because she had symptoms of COVID, even though she tested negative. So the system is a bit slow to get into place and then a bit imprecise in its execution. But we're learning. What this showed us is that WBE is actually a really useful approach for capturing data from locations that you might, on paper, presume is not going to be very effective. Why is this showing up? So the likelihood of finding an infectious case in a school, if you were to do the math, looked very unlikely. Because basically, if we think that 1 in 100 are going to be infected, and if you think that maybe 1 in 10 might use the toilet, you're talking fairly small numbers. And then if you're constrained by the amount of sampling or the quality of the sample that you're acquiring with an autosampler, which is on 1 minute, off 4 minutes, you're capturing only 20% of the flow. You're dealing with really small numbers. But despite that, we actually got really, well, as I say, 80% of the samples were positive by Christmas. So some of the assumptions that we had for schools in particular were clearly wrong. And we sort of allude to some of these in a paper that you can find online. This is what the whole school year looks like. I won't necessarily get into it more than just showing you that that the Christmas time, which is just a small fraction of the total data. So this is really everything after the Christmas period. And we looked at 2 genes, the N1 gene and an E gene, and they were mostly it was the N1 gene that was relied upon, but towards the end of towards the end of the school year, for some reason which we can't really explain, the E gene became a bit more useful than it was previously. This is just a particular school that was routinely testing positive. And that school was informed, and I don't think they were, unfortunately, they didn't have any systems in place for dealing with it. But it shows you that there are some schools that were chronically testing positive, where others were a bit more hit and miss. And it might have been hit and miss because the sampling was hit and miss. So we can't really speak to that, but when you're always hit, it doesn't speak well of the case rate in that school. So the next tier of work was in the prisons, and this is just a large amount of data from a series of prisons that we were doing surveillance at. And pretty much if there's a color other than gray, it meant that it tested positive. And then there's gradations of the amount of SARS that was found in the sample, but because we can't correlate quantity with infection cases or cases, the relationship is there isn't one necessarily. There might be one in some cases, but it's a bit of a debate as to can you derive a relationship between the amount of virus that you find in a near source sample to the amount of people who are infected. And it's because as you flush a toilet, you might just capture that toilet flush of the one infected person, and you might get a really good sample of that. And suddenly you have bucket loads of this virus in your sampler. But in other cases, you might get, you might miss either, you might miss a lot of flushes, or you might get the tail end of a flush and only get a tiny amount of the virus there. So the proportion of virus in your sampler is really more about serendipity than about numbers of people infected. But what this basically tells you is that you can use the same approach that we used in schools for the prisons. And here's some work that was done on the back end of the surveillance, was just to monitor it over time. So you can see that, yeah, as some of the prisons had enough data to show that they were going through a transition from really quite moderate infections to high infection load to then a much lower infection rate. And as I say, it's not proportional in any one case, but you can watch trends, and then you, because of the randomness of our sampling, the trend can be suggestive that the cases are dropping, and it does actually correlate with some of the sampling that they went in and did at these locations to confirm whether there was an outbreak or not, and there was. The right-hand side was variant detection. Basically, there was primarily only one variant going around at the time that we were looking. Right, so now transitioning into the viral, well, the second half of the talk, to be honest, which is on moving towards sensors. So population health surveillance can be done at an absolutely enormous number of locations, and I suppose it's our job to figure out, is there a good enough rationale to do that kind of surveillance at these locations? So I've shown you some data about wastewater treatment plants, I've shown you the network, and then the prisons and schools there, and care homes, which I didn't show you, but it has been done. Care homes are even more difficult because not everyone uses the toilet in care homes, because they have those adult nappies, which you don't get a sample from. So there are no shortage of issues that come up with almost everything. But there are a lot of locations that might offer you quite a lot of insight and potential protection. So critical industries like food preparation, food pickers. There are just a lot of things that we have come to take for granted that we definitely don't want disrupted. We call them key workers, and those are the people who we would very much not do well without. So there's an interest in making sure that if there's someone who comes to work who is infected, that we catch them very quickly. You know, you can of course swab them if the swab is sensitive enough and accurate enough, or you can do monitoring. Now, at the moment, monitoring is really expensive and it's not perfect. So there's a drive to try to make it closer to that, meaning inexpensive and closer to, you know, always getting an infected person when an infected person is there. So, as I say, there's no shortage of challenges, and this is really a very short list of a very long list. So one is, I spoke about the auto samplers. They're not fit for purpose. They often, almost always, go on a schedule, and basically as soon as you're on a schedule, you're inherently missing flushes, especially if you're near to the source. If you're further from the source, any flush event gets spread out in the sewer system, and the only negative to it being spread out is that it's by definition being diluted. So if there's only one person infected and it gets spread out, you probably will lose it if there's enough dilution. Detection limits are always an issue. There is increasing amounts of evidence that solids give you a better detection limit because basically you're pooing out the virus. So basically, if you're capturing poo, you're capturing where the virus originally was. If you're capturing the liquid phase, it's where the virus went after it's been flushed and through the sewer system. Temperature fluctuations, it can vary, you know, between sub-zero to, you know, well over 30 on a hot day, and these will impact the fate of the virus in situ as well as after being sampled. There are often no power options within a sewer system, so you basically need to rely on batteries, and then ice packs for the temperature as opposed to refrigeration. And then there's something called ATEX, which is basically a certification to have a mechanical object inside of a sewer system that won't make the sewers explode, because there's methane gas that happens in sewers. And so that's another constraint. These samplers, which is, I'm showing in the middle, they're quite big. They might not even fit in some manholes, let alone fit and be operational. And then there's transportation to the lab. It's probably one of the most costly parts of a whole service of providing wastewater surveillance at a national scale, is getting a sample from wherever it came from to a lab where the analysis is going to be done. And as I say, that's just a small list of those issues. Now, within every sample, you have, there's terminology here. I'll just hand it to you. Biomarkers, and biomarkers are basically the thing that you're trying to find, which would then help you understand something about the behaviors or the health of the population that you're doing surveillance on. One of the sort of longer term applications of wastewater epidemiology using biomarkers is to do illicit drug surveillance. And so what they would do is they would capture everyone's sewage, sample it most of the time at a sewage works, and then they would back calculate once they extract the chemical. They would know, when someone takes a drug, this much is metabolized and this much is excreted, and then you basically work your way back to the amount of drugs that are being consumed by the population. And so that's fairly well worked out, in fact, and it may not be publicized, but you can be sure that there are lots of governments who already do this. Now, something that's much more of academic interest, but actually is now starting to be of national interest, is the illicit drugs, I guess, which are basically the drugs that people are taking because they've been prescribed or they self-prescribe. And from that, you can actually determine compliance, which is, during the influenza pandemic, lots of people were prescribed Tamiflu for that, to combat the influenza virus. And I did a project during that time to measure the amount of the amount of Tamiflu present, and then you compare it to the amount of that was prescribed. And there was about 50% of the prescriptions were missing from the sewage, which was indicative of a population that wanted to know that they had the drug if they needed it, and about half of them decided they didn't need it. A bit like the petrol right now. You might have half a tank, but it'd be really nice to have a full tank. So that's just what humans do. So another biomarker is in the category of lifestyle. So we can find alcohol use, smoking. Then you could do exposure risks. So there may be metabolites from either of these, or metabolites from, or biomarkers from stress, from exposure to maybe industrial exposures to, or environmental exposures to pollution. And all of these can potentially feed into sort of an exposome, so that all the chemicals that you're exposed to. And I'm not saying that this is easy to do, I'm just saying that it's possible to do. There are microbiome understandings that can be generated from wastewater. So the microbiome of someone who eats meat is different from the microbiome of people who eat vegan or vegetarian. And so by interpreting the microbiome of wastewater, you can actually understand a bit of the dietary habits of a population. Obviously, the smaller the population, probably the more insightful it becomes. And then there's health and straight off health insights. So there may be diabetes biomarkers, stress biomarkers, nutritional biomarkers, mental health biomarkers. So the list is really long, and so I provide you a paper which goes into it in much more detail. And this is one of the figures in the paper, which I find really useful, but it takes a long time for me to get through these kinds of things because it's a bit of an assault on your eyes. But effectively on the bottom side of things, it's that things I haven't mentioned are antimicrobial resistance is a really important thing that is, well, it's a globally important human health concern, and being able to do surveillance for that in sewage is fantastic because it's almost perfectly made for it. I'll let you, find the paper and look at this at your own leisure. So there are two kinds of biomarkers. Here's another good paper which goes into this in excruciating detail. That author does everything in excruciating detail, but he's very good. And so it's endogenous and exogenous biomarkers. So endogenous are basically the stuff that comes out of you because it is a biological metabolite or from you, where an exogenous biomarker is something that you interacted with or is a product of your behavior. So it might be, the shampoo that you used in the morning is, has chemicals in it, and it's fairly easy to find that in sewage, or the tobacco that you smoke or the alcohol you drink. So most of the ones come into the exogenous biomarkers because they tend to hang around a bit better in sewage. Endogenous, it's no shortage of them. It's just whether A, do they biodegrade very quickly in sewage, and B, has anyone actually tried to look for them? So it may be that there's loads of them, but no one's actually tried to look for them in sewage yet. So there's a whole field of looking for these and seeing if they persist. So one of the use cases of endogenous is to track the relative time trends in collective health within a defined population. And then the second one is to compare the relative status of health across different populations. And this, you could probably think of many different use cases, but those are the ones from that paper. Another attribute of wastewater that is extremely important to try to get a hold of, and for sensors, it's critical that you latch onto as many of these as you can, because we're not really convinced that there's any one biomarker that is always good. So the more you latch onto, it's a bit like having more than one mobile phone mast that is connecting to your phone, because if that goes down, you don't have a signal, which I'm very worried I might have. So the more biomarkers you have, the more confidence you have about the data that, or the interpretation that you can generate from the data that you're going to generate. So normalization can proceed by two methods. Well, I say two because I had to, and then I added another, so now it's three, sorry. So one is straight census data. So once every 10 years, most countries will do a census, and that could be the data that you use. So if you know that the city of Oxford has 200,000 people in it, because that's what the census said, then you go and to the sewage works and you say, well, divide it by 200,000 if you have a 24-hour composite sample. That's probably the least precise. Wastewater treatment plants or the water companies themselves have spent a fair bit of time trying to understand the population that contributes to their business. And so they have their own numbers. Those are a bit more accurate, or probably very close to the real number. But to be honest, no one knows the real number because people are always moving around and you can't ever know the real number. But you can get increasingly closer. And another way that you can get closer to it is to measure proxies, chemical proxies. So some of these chemicals or biological markers or microbes can be used to derive a census or effectively a population. Crasphage is being used by a number of people. 5-6 is just another bacteriophage that is associated with things that we can back calculate to. Mitochondrial DNA is another really strong candidate for a biological marker, because that's coming from the host itself, the human, as opposed to crasphage, which is coming from the basically from the growth on bacteria that are in your gut. There are chemical markers that have varying degrees of stability. So creatine is one, and the long list of other drugs like carbamazepine is it's widely used in society, and it doesn't degrade very easily at all. So basically, you can back calculate to a population size by the amount of carbamazepine. And there are other drugs, not drugs, but chemicals like artificial sweeteners that you'll find in squashes and things like that, or fizzy drinks. A lot of those can persist, and so they become good markers. So, of course, biomarkers are subject to, their biodegradation. So the better biomarkers are the ones that don't degrade, or they degrade extremely reliably. As you could imagine, not degrading is better for this purpose, but it's not great for the environment, because that means the thing is just going to be there forever. And biomarkers, obviously you want them to be of the right order of magnitude, so that you don't want it to be too low, and you want it to be sensitive to the population. You don't want it to be sensitive to how heavy someone is, or whether they have another condition. You would prefer it not to be age specific or gender specific or even diet or ethnicity specific, but as it turns out, most things are. And so what you need to do is get a handle on how important those are, and then apply potential correction factors for population, the demographics that you're studying. In many cases, you might not need to, because the numbers are imprecise enough that imprecision might be trivial by comparison. So it really needs an understanding of the kind of numbers you're generating. Right. I think I'm getting close to the end. Is post-pandemic pathogen priorities. So we're moving into a time where we're not post-pandemic, but we're thinking about it. And in fact, we can't ignore it, because life has continued on. We've gone back to work. A lot of people have actually gone back to the office. And we will be getting all the regular pathogens that we always got every winter. And those include influenza, norovirus, respiratory viruses. And so those are high priorities for incorporating into the pipelines that we're currently running for SARS. Some of those will be RNA viruses, so they can just slide in quite nicely into the same pipeline that you might already have. But those pipelines, usually end off in a qPCR kind of analysis. But there's LAMP approaches, and there's quite a number of new approaches that have emerged during the pandemic, because there's so much interest in detection. Polio has emerged from nowhere. Well, not literally, because actually we know exactly where it emerged from. There's several tens of planes that left Afghanistan not that long ago. And there's a risk that the people who got on those planes might be carrying polio. And because Pakistan is and Afghanistan are two of the places where polio has not been eradicated from, thus it becomes an issue here. And I'm not sure, but I've been led to believe that polio's vaccination is not as frequent or assumed as it once was when I was young. So there may be a susceptible population, which would be absolutely horrible. So there's a renewed interest in polio surveillance. It still is ongoing in the UK, but not at a national scale. So it may be that places where people settle from these locations might be under surveillance for polio. It might be a sensible thing to do, just to make sure that there isn't something that's emerging that needs to be done, or just go in and vaccinate everyone. Anyway, I'm not here to solve the world's problems on that yet. So AMR is that global problem that is really quite well suited for wastewater surveillance. So AMR is anything that you would find in the bacteria and fungal targets, and it hasn't been I mean, there's loads of research on AMR in wastewater, so that's easy to show. Some of these slides I'm showing norovirus. There was basically virtually no norovirus for most of the year on the x-axis, and then as we get closer to current, it goes over the blue line, which the blue line is your 5-season mean. So, but it may, you know, be in line with, or it may exceed our expectations. Time will tell. And on the right-hand side is influenza-like illness. And again, there was virtually no flu in the recent year, but we're expecting that not to be the case again. When you set up these kinds of surveillance, one of the things you do need to ask yourself is, what can you do with this information once you collect it? Can you make it impactful? Is it the kind of information that's just good to know? So it's confirmatory. Maybe the expectation is that there is no polio in this country more than there was at any other time, and it'd be good to know that that's actually true. Or is it corroborative, where, you know, you do have people showing up in the hospital with something, and you just want to get a sense of, is this a bigger problem than we think? And the last thing is, this came out just about a day ago. It's the application of effectively wastewater epidemiology into the environment. So you may have seen this in the news in the Glastonbury surprise. People use drugs, and those who don't use the portaloos use the river, effectively. And so someone cleverly, well, not just someone, there's the paper there, a number of people from Bangor actually, went and took samples from a sort of a control river, which wasn't as controlled as you think a river should be, and then the river that was downstream, well, upstream and downstream of the festival, and they found bucket loads of drugs, and in fact so much drugs that they were actually trying to figure out what kind of impact that might have on the wildlife, because it was enough that you would think there was probably going to be one. So it's a really nice example, unfortunately for the world, but it's a nice example of everything I've been talking about, of how you can use wastewater epidemiology to identify issues, and then, you know, there may be mitigation measures that are going to happen next year because of this knowledge. And there may be also people looking at the wildlife to see, is there really an impact or not? Whereas before we wouldn't have looked. Okay, so I would say final thoughts, but I think I'll just leave it up there for a bit and say I have about 8 minutes before you have to go off to your 1 o'clock appointment. So I'll just say thank you very much for your time. And all of you who might be interested in or developing sensors, there's never been a stronger need for sensors than there is now. And there are never more targets for sensors than there is now. So you're in a fantastic place, and we need your help. So thank you very much.
Well, thanks very much, Andrew, for this fascinating talk to give the overview of WBE, especially in the current progress in the UK, covering near source tracking and also other biomarkers, and also the future trends for post-pandemic pathogens detections. So we got a few questions from the audience. So first, comments. It's really good to see this initiative and the fact that it will fit into students' education and work with industry. So try to inform environmental regulators such as SABR and the EA. It's very positive, and hopefully it can be well received. I come to the next question. Yes.
John?
Yes, please, John.
Yes, thank you very much. Thank you, Andrew, for a fascinating talk. There is a lot to think about there. There are a couple of issues that come to my mind, and that is when you mentioned the costs of taking samples and transporting samples, and therefore the benefit of in situ, if you like, sensors. I wondered, when one takes samples, whether it's possible to preserve them or an extract of them to form a sort of reference library for future analysis if required, that kind of application. And the sort of second part of this is, in relation to the development of, for example, in the UK, analytical capability and availability to apply the science? In other words, longer term, how is the science being developed, and what type of informatics are being used to capture and interrogate and to develop artificial intelligence in relation to this kind of data? Because it's, as you say, very varied data that has potential applications across a very wide spectrum of environmental public health challenges. So just really the one about sample stability and reference library, the one about building capability, and the one about analysis of data and intelligence.
I might need you to go through each one. So the first one is sample stability is identified as an issue for until we have an in situ sensor, or in addition to, if the desire is there. There are efforts to try to stabilize samples for analysis, and we probably will be in a better place when it comes to that kind of a question quite soon. I can point you to Zhugen in about 3 months' time. He'd probably be able to tell you a bit more about it. So that's a one to watch. And with regard to the artificial intelligence, so how do you deal with all that data, and how do you make best use of it? AI, or any machine learning, is going to be really useful. There are discussions about that kind of approach, and in many ways it requires an enormous collaboration between data holders and data jockeys to basically pull that off. And so it's one of those things that you should be pushing for if you don't see it. And to be honest, right now it's not the norm, but it probably will be the norm once the amount of data that we collect continues, and then not only continues but grows. We're going to have an automated way to interpret that, and there's so many opportunities to bring in not just chemistry data but just human behavior data. So it may have been that the sewage works at Glastonbury, we would have been able to identify these things using a machine learning approach, because if you scrape the data off the web and the web tells you that there's a concert in Glastonbury, it already knows what's going to come. But it can do that for everywhere. So there's a concert at the O2, it's going to scrape the data from that, make some predictions as to what it's likely to find. If you're interested in illicit drugs, and you don't bother looking for it on the days that there are no events, but then you start looking at, so you can have a really smart system that is very responsive, but it requires AI. I mean, or just this kind of integrated data, and I think it is what we're aiming for, meaning as a society that is the direction of travel, but it's we're so close to the beginning. So I wouldn't say we've gotten very far.
Thank you very much. Thank you.
Thank you, Andrew and John's question. And I've got another question, which is very interesting. Outside of disease monitoring, can you see a role for democratizing this kind of monitoring, perhaps through citizen science in a broader environment?
Yeah, so that's where a lot of the power is going to be in the future. So as soon as you clever people make sensors that are cheap and easy to use, then you have basically empowered every country to be able to have their citizens do the surveillance that the governments typically can't afford to do. So it's really expensive to do monitoring using current up to present day kind of monitoring methods. But if all of a sudden you have really motivated people on different stretches of river, have a really vested interest in the quality of their river, they will go out, they'll take a sample, they'll swab it, whatever the sensor requires them to do. They'll come up with a number. It might be something that there was an automated process of getting that data into a cloud, which then has action, it may be something that the regulators work with to action, or it might just be the evidence base that they can go to someone and say, this is not right, we need to do something about it. So you're empowering the local people to generate the data that is the evidence base for them to then, cause action to happen in some sort of intervention. And this is independent of government. This can all happen with, some of these sensors could be pence per test, and that's extremely affordable, and it's also scalable, and there are a lot of motivated people in this world who really want their backyard to look really nice, and so they will go out there every day and take these samples. So I think we can never underestimate the population's willingness to do this. You just have to give them the tools, and the tool that's missing is a sensor.
Yes, I think it's a very fascinating areas. It's just being a lot of data driven to push some new insights in these areas.